Hi guys, how are you? I hope you're good, not feeling too overwhelmed with metabolism. So I expect that you turn on this video after you watch those videos that I posted the playlist about cellular respiration. So, from what you saw in that, let me get my pen here. From what you saw in that video about cellular respiration, do you agree that we have some ATP being formed in glycolysis? And what is glycolysis, right? Oops, sorry. What is glycolysis? Glucose is being broken down. One molecule of glucose is broken down into two molecules of pyruvic acid. Okay, so we're just now talking about this part. Okay, and that generates some ATP and some NADPH. What's the NADPH? So I'm just here talking really quickly about what you just learned, okay? Just an overview. What is NADPH? NADPH is that electron carrier, is a cofactor that helps enzymes in this metabolic process to work. And one of the things that it does is bring electrons. How does it bring electrons? Remember that you have hydrogen molecule can be divided into hydrogen ion plus an electron. This is the electron that is being carried by NADPH here. So then we move on, right? And then pyruvic acid is transformed enzymatically into acetyl-CoA. Acetyl-CoA combines with a molecule that you have here in the Krebs cycle and creates a 6-carbon compound. That compound is going to be broken down during this cycle, and as it loses carbons, you release carbon dioxide. Is that the carbon dioxide that we exhale when we breathe out? It is. So what else do you need to know here? You also generate ATP during this process. So you have ATP being generated here, right? During glycolysis, ATP being generated during the Krebs cycle. What else does the Krebs cycle do? Generates carbon dioxide because it's breaking parts of that molecule that was formed here down. And it generates more NADH and more FADH2. These two molecules are also, the one I already told you, the NADH and the FADH2 are both carriers of electrons. Electrons are carried where? To the electron transport chain. What is the main purpose of the electron transport chain? To generate a lot of ATP. What is ATP again? It's the molecule that contains the energy that we need for all the other processes. So we're breaking down a molecule of glucose to generate energy for all the other things that we need to do in the cell. What are these little purple circles here? These are the acceptors of those electrons. So electrons jump, boing, 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 boing. And as they jump, you have that gradient of hydrogen ions changing, and then you watch the video, right? And the video probably talked about mitochondria. So in the mitochondria, these hydrogens that are being formed here, these hydrogen ions that are being formed here, are going to be formed in the intermembrane space of the mitochondria. What if this is a bacterium? Where is this being formed? So I'm going to just draw here. If this is a bacterium, you have the electron transport chain happening in the plasma membrane. And as electrons go ping, 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 ping through these molecules, these electron acceptors, hydrogen, moves out of the cell. Hydrogen ions move out of the cell. And that causes what we call chemiosmosis. So chemiosmosis is the fact that you create a gradient of hydrogen ions, which is more concentrated out of the bacterial cell and less concentrated inside the bacterial cell. For what? Remember, in the end, here, in the end of the electron transport chain, you're going to have an enzyme that is called ATP. Oops, hold on. Back. ATP synthase. What is the ATP synthase? It's a pump. It's an enzyme that works like a pump that makes ATP. Makes a lot of ATP that we talked here is made in the end of the process, right? It's made here. So just make it bigger again. So what are we talking about here? We're talking about ATP synthase starts to spin, and what gives the ability for this pump to spin is the fact that you have a lot of hydrogen ions present. So these hydrogen ions are what activate the ATP synthase to make what inside the bacterial cell in this case? Lots of ATP. Okay, so you generate between 28 and 32 molecules of ATP just in the electron transport chain, which is a lot more than here in the glycolysis and here in the Krebs cycle. Okay, so it is a very high yield process when it comes to molecules of ATP being formed. Okay, so now if I erase this mess, just so you can see better the other side of this table, what do we have? So now let's write with another color. Fermentation. What is fermentation compared to respiration? Well, you're going to say, well, we don't do that. We do. Our muscles, our skeletal muscles ferment. You know when you go to the gym and you work really, really hard and then next day you feel sore and you're like, oh, I heard something about lactic acid accumulating in my body, in my muscles. Yeah. Where is lactic acid coming from? You used all your oxygen because you were hyperventilating and exercising really intensely, and it gets to a point that you start to do an anaerobic process that is a fermentation. So you produce lactic acid in the muscle as a way to make energy from glucose during that process, so you have enough energy for your muscles after you consume all the oxygen there. Okay? So, but now we're going to talk about bacteria. So what bacteria do? Remember how some bacteria can do aerobic respiration and some cannot. Bacteria that are facultative anaerobes, I'm going to write here, facultative anaerobes, they mostly do fermentation, and if there is some oxygen present, they can do the aerobic respiration. So they can do this, but most of the time they're totally okay just doing this. So why do they do that? Why do they ferment? To generate ATP. That's our whole purpose here. We're breaking something down, which is glucose, to generate ATP, okay? And with that, you break it down into pyruvic acid, you have a glycolysis in the beginning of fermentation, and then that pyruvic acid is going to form fermentation end products, which end up being the molecules that will receive that oxygen in the end. So who is not the oxygen, the electrons. So who is the final electron acceptor here in the respiration? Oxygen. That's why I said oxygen. I got confused. Who are the final electron acceptors here? So remember how electrons are being carried by the NADPH? Where do they go? To whatever end product is being formed here. Which are the end products of fermentation? Alcohol, propionic acid, acetic acid, ethanol. Remember, even yeast can make ethanol. Usually yeast make ethanol out of fermentation. That's how we have beer, and that's how we have bread, right? Bread is when yeast ferment the sugar present in the bread, in the starch, and produce alcohol and carbon dioxide. Sorry. The carbon dioxide is going to make the bubbles in the bread, and in that case, the alcohol is going to evaporate when you bake the bread. Okay? In the case of the beers, mostly you're more concerned about producing the alcohol and using that alcohol for the beer, right? But it's the same process. It's fermentation by yeast, and you need sugar for that, right? Sugar is being broken down. What else can you form? I said acetic acid, propionic acid. You can form other types of acids or acidic substances and other types of alcohols that we could mention, but that are different ones, okay? We're going to use, for example, butanediol is an alcohol that is formed during fermentation of some specific types of bacteria, okay? So there is a slide in the book. I'm trying to think if I have that slide here. Let's see. That tells you all the byproducts of fermentation. Yes, I have it. So, see, lactic acid, ethanol, and CO2, and it shows you which organisms would produce the different things. So the Saccharomyces, which is yeast, produce CO2 and ethanol. The Streptococcus, Lactobacillus, and Bacillus produce lactic acid. Propionibacterium produce propionic acid, important for production of some types of cheese. Acetic acid, vinegar. Butyric acid by Clostridium, butanol, acetone, isopropyl alcohol by Clostridium. Salmonella and E. coli produce all types of different acids. They make what we call mixed acid fermentation. And Enterobacter can produce other different types of things, including acetoin, which is something we're going to learn how to measure in the lab through there is an assay in the lab that is called indol. So you're going to learn that later. Anyway, going back to where we were, which of these two processes, now I have here, never mind, that's just a summary of the glycolysis. So if we go back to where we are, which of these two processes do you think produces more energy? Totally here, more energy, right? Which one of these two processes is anaerobic? Totally fermentation, right? Anaerobic. How could this be anaerobic if you use oxygen? It's totally aerobic. The oxygen you inhale is here because it is the final electron acceptor, right? Cool. So this is my overview for you of what you just heard for the second time. You do not need to memorize or learn all these byproducts of glycolysis until we get to pyruvic acid. Don't worry about that. Actually, don't worry about any specific name. Like citric acid is the first one in the Krebs cycle, and then you have all these other acids being produced after the transformations that happen during the Krebs cycle. Don't worry about those names. And like I mentioned, this is the ATP synthase that through the action of the proton, through activated by the hydrogen ions, is going to spin and form ATP, right? So this is the general equation for aerobic respiration, right? You have glucose plus oxygen with ADP and phosphate generating carbon dioxide, water, and 38 ATP. There is also a process called anaerobic respiration where this oxygen is replaced by other electron acceptors, for example, nitrate, sulfate, and bicarbonate. Okay? And the products after this reaction is over is not, so instead, so here is instead of forming CO2, you're actually forming nitrate, nitrogen gas, water, sulfuric gas, and water, methane, and water. Okay, so these are still catabolic reactions. It's an anaerobic respiration. You're still breaking down glucose, but because there's no oxygen present, you are forming other end products and using other electron acceptors. That's all you need to know. Okay. All right. I think what we have left here, we talked about fermentation. Yeah, we just need to talk a little bit about lipid and protein metabolisms. And yeah, and I'll tell you what else it's in my mind in a minute. So proteins are broken down into amino acids, right? And if you look at this, that's not what I wanted to show you. Hold on. So there are different places in the cellular respiration process where amino acids can be transformed into molecules that are part of this metabolic path. So you don't need to know the names, but amino acids can be broken down into substances that end up producing pyruvic acid or acetyl-CoA or some of the components of the Krebs cycle. Does it make sense? So you could use protein when you don't have carbohydrates, sugar, to generate energy because of this ability of the byproducts of protein degradation to produce byproducts from this metabolic pathway that is the aerobic respiration. The same way lipids, when they're broken down into glycerol and fatty acids, can be transformed into, for example, glycerol is transformed into glyceraldehyde triphosphate. Fatty acid is transformed into acetyl-CoA. So you can use lipids also to produce ATP. When do you do this? You know that Atkins diet where people don't eat any starch and any sugar and all they eat is protein and lipid? How do they get energy? Through this process. So not having sugar is still okay, I guess, for some time, for you to survive and have your metabolism and your ATP production kicking in. What is the problem with that? When you don't use this and you use only amino acids and fatty acids and glycerol, you're going to increase the pH. Decrease the pH of your blood, and you're going to produce acidosis because of this, the production of ketone bodies, ketone acid, which are substances that increase the pH in the blood. And that can cause other issues because changing pH in the blood is not a good idea. So that's why it's not ideal that you do not consume any sugar and use only amino acids and lipids. But for example, individuals that are diabetic, they need to be in a diet like this, and they end up having a lowering in their blood pH because of that. Okay, but that's a necessity, right? They cannot eat sugar because they cannot transport it to their cell. Okay, so we learned that proteins and lipids can also participate in the cellular respiration process. Now, I'm going to show you an assay. I want to focus more on this assay, which is called fermentation test. And what this is that you have a medium that has a substance that is called phenol red. And what phenol red does is it's a pH indicator. So when the pH of the medium is acidic, what's going to happen? It's going to turn yellow. When the pH is alkaline, when the pH in this whole broth, in this whole medium, is going to be pink or red, okay? What else do you know what this and what does that really mean? Remember we said the fermentation can produce acid. So if you inoculate a bacterium in this medium that is able to ferment whatever sugar you have in this medium, these are carbohydrate broths. So they have a lot of sugar. Which sugar you put, you can put different sugars, and then you know, let's say you put just lactose and your bacteria is a good lactose fermenter. What's going to happen? That bacterium is going to eat the lactose, produce acid. What's going to happen to the medium? It's going to turn yellow. What if the bacteria does not like lactose, does not ferment lactose? What's going to happen? It's going to continue to be pink because it's just going to continue to be alkaline, or it's going to go from neutral to alkaline because the bacteria doesn't like the sugar and starts to eat the amino acids in this medium, produces ammonia, and ammonia is alkaline. So it gets even more red than normal, gets like a pink color. So what I'm telling you here is that this is a fermentation test. So whatever bacteria you put in, you can test if the bacteria can produce acid because it fermented that specific sugar that is in the medium. All right, I am going to stop here and I'm going to leave you with this image and I want you to think about what this is. I think you already learned all this. So this is just a review of the ATP production and what are the requirements. And I have a third video in this playlist, which is about the energy, the nutrient requirements. So you have 4 different types of organisms: chemoheterotroph, chemoautotrophs, photoheterotroph, and photoautotrophs. In the next video, it explains to you what's the difference between these 4 organisms. So please watch the next video and let me know if you have any questions, okay? Bye-bye.